All right, so we could get started. So you guys already know my name, right? So I'm Moses, right? All right, so today we're looking at precision and personalized medicine. All right. I thought that the title was a little bit awkward because the lecture is just dealing about cancer, right? So basically, like how you generate cancers, like through mutations. What specific things get mutated, and then later on in the lecture, the professor focused on colorectal cancer specifically. So basically, the lecture is about cancer, what leads to the development of cancer, and then at the end of the day, he starts talking specifically about colorectal cancer. All right. All right. So beginning off, all right. So when we're looking at cancer, all right. So you're going to start off with normal cells, all right. And then with this normal cell, you're going to have a mutation that occurs. All right. And usually that first mutation that occurs allows the cell to rapidly divide. All right. So this is hyperplasia. Right. So with hyperplasia, you're still having normal cells. Right. Those cells are still like normal. They're not abnormal. Right. That's hyperplasia means like too much division of normal cells. All right. That's what that is. So with the first step of generating that cancer, all right, you're gonna get a mutation, and that first mutation usually leads to hyperplasia, which means that the cell is rapidly dividing. Okay. All right. So here, I'm not sure exactly where we're at here, but you guys can see it's some kind of epithelium, right? And you can see the cell right there, right? Make this a little bit smaller. Okay, so here goes the cell. All right. So what's going to happen first? You get a mutation, right? That's going to lead to abnormal division, but the cell is still like a normal body cell, right? So you get hyperplasia, all right? So you have all these cells right here. That's the first thing that happens. Okay, now with that first mutation, all right, you get another mutation, all right? Now usually this second mutation could also still cause more like division, hyperplasia, right? Um, but it could also lead to a change in the shape and morphology of the cell that is being created. Notice that the hyperplasia did not lead to a change in shape or the morphology of the cell, right? But as more mutation starts to occur, the normal cells are going to start to become abnormal, and they're gonna take on they're gonna start having aberrant features. All right. Okay, so here we go right here. Okay, so we start off with a cell, right? Normal cell, right? It gets a mutation. It starts to rapidly divide. The cell is still normal, right? Then you get another mutation, right? So let's just say this is mutation one, mutation two. Then you get another mutation, and now you get dysplasia, all right? So dysplasia means that the cells that were now normal are now taking on abnormal characteristics, all right? And you can kind of see that, okay, whereas I'm saying these are a little bit, you can see the color here. They're almost like, it's almost homogeneous. But then you can kind of see like, okay, you got a blue thing here. You got like some color changes going on here. So you're starting to see abnormal characteristics in the shape, morphology, see the color, content of the cell, stuff like that. Okay, dysplasia. All right. So we start off with a cell, we get a mutation, we get hyperplasia, we get another mutation, leads to a change in shape or morphology. All right, then you get still other mutations. All right, and these other mutations can confer characteristics such as the ability to penetrate the basement membrane. All right, so as you can see here, so all these epithelial cells, they are sitting on a basement membrane. All right, and you can see it here, right? It's a little bit lighter, right? This is your basement membrane. All right, so as we're getting these mutations occurring, it's going to get to a certain point where um, these abnormal cells are going to be able to penetrate that basement membrane. Okay, so below that basement membrane, you have blood vessels. All right, so you see this blood vessel right here, right? And so the first step for that cancer to start to metastasize is it has to break through that basement membrane, then get into this connective tissue area here. And then within that connective tissue area, you have your blood vessels and you have your lymphatics, right? So some cancer cells are going to travel through the blood vessels and some are going to travel through your lymphatics, right? In this figure here, this one is traveling through the blood vessel, all right? Okay. So other mutations such as being able to penetrate through the basement membrane, all right? Being able to invade other tissues. So once you break through that basement membrane, all right? Now you have your connective tissues, your fibroblasts, right? Now you're invading other tissues, right? So the epithelial cell was supposed to be on the surface. Now it's no longer on the surface. Now you can actually find it in the connective tissue area, all right? And then you have so ability to draw like blood supply. So as you can see here, clearly this is a blood vessel, right? But now it's kind of feeding into this cancerous mass because the cancer has acquired a mutation that actually allows it to draw blood supply to itself. All right, cool. And let's see here, and then ability to enter into the blood vessel, right? So basically, when you're looking at the story of what is a cancer, you're looking at series of mutations, all right? So when you look at cancer, if you say something is cancerous, all right, what you're saying is that there were a series of mutations, right, that created this cancer, right? Is this clear? I don't know if this makes sense. Yes, maybe. I'm getting strange looks.
No, it's good.
Okay, cool. All right. So you get a series of mutations that results in malignancy. Now another thing here, that you can see here is that when you're having this cancerous mass, so some of the tissue actually becomes necrotic, so like meaning like some of it actually dies because there's not enough nutrients to properly, go through all the cells. So you start to get like necrotic areas, right? Like tissue, you get tissue death, all right? So you can see that here as well. Okay, just in case you didn't get this the first time, I'm gonna repeat the same information again. Okay, so in this figure here, we have acute inflammation. Okay, so essentially if you have an injury or an infection, and if it results in an acute inflammation, the cells that are damaged will eventually start to get repaired. So basically your body is going to try to regenerate those cells. Okay, 1 second. Okay, so let's get back on track here and make sure this thing is actually working. Okay, so it is working. Okay, so what I'm saying here is that if you get acute inflammation as a result of injury or infection, what happens is that after you have that injury or infection, those cells that died will start to replicate, all right? Now, as these cells start to replicate, now you have the possibility of acquiring a mutation that could lead to progression to become cancerous or whatever. So when you have the cell and, it's trying to divide, it can acquire a mutation, right, that will confer it with the ability to divide more or faster than it should. So here we have this cell right here. So it's starting to divide faster than it should. And what did we call this? Hyperplasia. That's right. Okay. Now you guys, okay, so the mutation could just be, again, it could come from different sources. So it can just come as a natural process of, this, just natural mutations that can occur. Or it can be like environmental, so like, radiation, UV light, carcinogen, stuff like that, that can also lead to the mutation occurring, leading to this hyperplasia, okay? So here it's showing hyperplasia, well, it's showing mutation in the epithelial cell and also the stromal cell. So this is like your connective tissue, right? Right. So this is like fibroblasts. All right. Okay. So from here we get another mutation, right? And it leads to further proliferation. But now, the way that this cell here looks is a lot different from here, right? So what would we now call this?
Dysplasia.
Okay, dysplasia, right? Okay, so we went hyperplasia, dysplasia. We're yet still getting more mutation, right? And so we can kind of see that, it's kind of drawing some of the blood supply here. Well, I don't really see much here, so it's just getting a little bit worse here, all right? All right, so the story of cancer is a story of a series of mutations, all right? Yet again, I'll repeat it again. Okay, so you get one mutation, right? You get another mutation, right? Then you get another mutation, and then you get other mutations, and then you get that malignant tumor that you're looking at. Yeah.
Okay, so that's an example of clonal selection that he was talking about? That one? Is that it?
Clonal selection. Where do you see clonal selection?
I don't know. You mentioned that in the lecture, and it was on that slide, is that it? Like selecting for the malignant clone?
Okay. Well. Okay. So, normally when you have a cancerous mass, all right. So, it all started from one cell. So one cell started that whole hyperplasia, right? So you get all these cells that were like the first cell that caused the whole thing to occur. So by clonal, I think it's referring to that first cell that started the whole entire problem. But I'm not exactly sure what exactly you're referencing.
I think this slide, because it wasn't
Oh, okay.
the PowerPoint that we had.
Okay, clone. Yeah. Okay. All right. Thank you. This is actually pretty helpful. Thank you. Okay, so okay. I don't know. Let me just draw. Okay, so these are a bunch of cells, right? And let's just say this one right here acquires a mutation. And we said that first mutation was what? Hyperplasia, right? Okay. So now we have, and let me make this a little bit different so it stands out a little bit more. One second, you guys give me a few moments. Okay, all right. Let's get back on track. Okay, so what we're saying is you get a first mutation, hyperplasia, right? So you get a bunch of this first cell here. All right, this cell here would be the what you call it? This is the cell that started the whole thing. All right. So all of these cells here came from this guy here. So I think that's what's being referred to by the monoclonal, like they were all derived from one one clone. They're a clone of one particular cell. I think that's what's being referred to. Like that the cancer itself was derived from one cell. So they all resemble or have maybe some resemblance to that particular one cell. Okay, cool. Did that help a little bit? Okay, cool. Okay, so we talked about the process of generating the cancer, all right? So, but now, like, what exactly got mutated, all right? So, what genes or what things actually got mutated, all right, to bring about the cancer? So there's two things that we're gonna look at. Um, all right, so there's two key things that we're gonna look at that will get mutated, all right? So that's gonna be your proto-oncogenes, right? And your tumor suppressor genes. All right, so does anyone know what a proto-oncogene is? Just putting it out there. Does anyone know what a proto-oncogene is? Okay, yeah. Okay, so there are genes that promote cell growth and cell division. All right, so these proto-oncogenes, they're promoting cell growth and division. Okay, now we have these genes, they're promoting cell growth and cell division. Well, if they're abnormally active, then we get too much cell growth and we get too much cell division, and that promotes the cancerous state. Right? Okay, then you have your tumor suppressor genes. All right, so it's basically logical. So these genes are trying to suppress tumor generation. So these are like your the genes that are going to be promoting apoptosis. So like they're going to be trying to eliminate cells that become abnormal, all right? And they can also prevent progression into the cell cycle. So they can prevent cell cycle, or they can regulate cell cycle. These guys regulate cell cycle, all right? And they promote apoptosis. All right. So if the genes that are normally supposed to promote apoptosis, if they're no longer there, then the cell, when the cell becomes abnormal, then that abnormal cell can now proliferate because we're not inducing apoptosis. Everyone knows what apoptosis is, right? Okay, so if we don't have these genes present that normally take out the abnormal cells, then we can get proliferation of abnormal cells, and that promotes the cancerous state. Okay, so the tumor suppressor genes, all right, they're going to be promoting apoptosis when they recognize that something is wrong. So if your chromosomes, something is wrong with your chromosomes, stuff like that, these genes are going to, or the products of these genes are going to promote the self-destruction of the cell when something goes wrong. So if they're not there, then something is wrong, but then the cell is still left to do whatever it wants. It can still grow and stuff like that. So it promotes the cancer. It promotes the dysplasia, right? Cool. All right, so what we're going to do, we're going to dive into this, starting first with the proto-oncogenes, all right? Then we'll look at just a few of the tumor suppressor genes. Okay, so again, just repeating what was already said. What are proto-oncogenes? They are genes that promote cell division and growth. All right. Now I think, let me just give you guys a quick idea really fast here. Okay. just say. Okay, so for your proto-oncogenes, they're gonna correlate with this little pathway that I have drawn here. Okay, so here this would be representative of a growth factor. All right, and then this here will be representative of your growth factor receptor. And then this here will be representative of the signaling cascade. And then this little ball here is representative of your transcription factor. All right. And then, yeah, and it goes to the nucleus. Okay. So things can go wrong, all right, at any of these points here. Right. But you can see all of these guys. Second, this is not helping me. Okay. I guess I got it. Okay. Okay, so you can see all these guys as proto-oncogenes, or they are products of proto-oncogenes, right? Because they're proteins, right? They're products of proto-oncogenes. Okay, so let's just think about this now. Okay, so if you have a growth factor, all right, so if you produce too much growth factor, then that's going to be causing the cell to what? It's going to cause a lot more divisions, right? So if you're getting a lot more division, that's a problem. That promotes cancer, right? Now, if you have too much of the receptor, that's also a problem, because now, like, it's very easy to detect the growth factor, right? So that leads to more division, right? Or if some of the signaling, intermediates here, if some of them are just for some reason they're just on, right? Then they can go and activate the transcription factor, which will promote, upregulate those, those segments of the DNA that leads to these products, right? So that's also a problem. Or if the transcription factor is, somehow on, that could also lead to the same thing. So essentially, anywhere along this pathway, something can go wrong, which will promote cell growth, cell division. Does this make sense? All right. So when you're thinking about proto-oncogenes, you're thinking about something within this kind of like cascade here. All right. Okay. Does anyone have any questions so far? Is there anything? Yes, sir.
So are we supposed to assume that for all the proto-oncogenes, like, is it just like a kinase cascade? Like, should we like know?
I will go a little bit into detail. Now, my understanding is not 100% complete. So, again, I say I'm a student, but we will see a bunch of different examples. And hopefully by seeing the examples, that would help to maybe give you an idea of the possibilities, right? Sorry, can you see me? Yeah, that would give you an idea of like the possibilities. So, but usually, at least from the ones I've seen so far, it's with the growth factor on the growth factor receptor. Usually the ones that I've seen. Cool? All right, so now going into the proto-oncogenes. All right, so let me ask you guys a question now. Okay, so can you guys give me an example of a proto-oncogene? Growth factor. Okay.
transcription factors,
Transcription factor. Okay.
cascade factors.
Okay. Okay. All right. Okay. So another question. So what is
Dysplasia.
wait. Just one person. All right. I just want one brave person here. All right. Go ahead. Go ahead, dude.
Alright, isn't that just the change of shape in cancer cells that it just disfigures and the morphology changes?
Exactly. Okay. Good. Good. All right. And so question for you again. Okay. So then what is hyperplasia then?
No change.
Okay. Okay. Go ahead. You can.
There's no change in shape, but you do have like a couple of cells.
Okay. Okay. That's exactly right. That's exactly right. Okay. Cool.
It's self-proliferation, but no change in actual shape.
Yeah.
Can the people on the sides like move, scoot over so you can like all, because there's people outside that can't
I'm
see.
actually recording this, so I'm gonna record this and I'm gonna post it. So like right now, everything we're saying is being recorded.
Yeah, it's gonna post it.
All right. so does anyone else have any questions or? Okay. Okay. Okay, so now we're going specifically into the proto-oncogenes, right? And we already established what the proto-oncogene is. All right, so for your growth factor, so this is what was mentioned, and I'm taking this directly from the slides. So I just try to organize this in a way that makes more sense. So for the growth factor, the one that was specifically mentioned in lecture is your platelet-derived growth factor, all right? So when you have the cis oncogene, all right, cis oncogene or whatever, cis oncogene, when you have that, you have overproduction of your platelet-derived growth factor. But if you have too much of the platelet-derived growth factor, that's going to promote the cancerous state, right? Cool? So if you have too much platelet-derived growth factor being produced, right? So it's basically like you're like stimulating the cell and you're telling it, you know, divide, grow. You know what I mean? So if you're producing too much of this growth factor, then you're promoting the cancerous state.
What did you say about the cis oncogene? Yeah.
Okay, so I guess so when you have that cis oncogene, it leads to the overproduction of the platelet-derived growth factor.
So,
Hmm?
so overproduction goes to overproduction.
So cis oncogene is the mutated version of the PDGF causing overproduction
Okay.
of cancer.
I'm not 100% sure. I took this from the slides. So there is a connection between cis oncogene and overproduction of PDGF.
I have a question. But you don't know where. So
Yeah.
if something is inhibited, like tumor suppressor, whatever, what kind of oncogene would that be?
You say that if the tumor suppressor gene is inhibited?
Yeah.
Okay, so remember we just established that the mutations that we're getting, at least the ones that I saw in the lecture, is a mutation of either the proto-oncogene or the tumor suppressor gene. And now within the proto-oncogene, we establish the possibilities within the proto-oncogene. So the tumor suppressor genes is a separate class, a separate category, right? That's what we were establishing over here. All right. Cool. There are yet other classes, but in terms of like the scope of the lecture, right? I'll try to keep it within the scope of the lecture. Okay. Is everyone good on this so far?
Question.
Yeah. Sorry. Go
So
ahead.
the platelet-derived growth factor, you having more, is that like a cause of like something environmental, or why would you have an excess of that?
Okay. So it can be as a result of many things. I'll just reference you again to this. So again, it can just be a mutation, and you know, carcinogen. It can be genetic. So there's a lot of different sources that leads to the mutation.
So it's nothing in particular for the cis oncogene.
I'm not exactly sure what was the cause of it, but yeah, something did cause it, you know, and then it mutated. Right. So we talked about the growth factor, specifically the one that was.
What's cis oxygen?
Well, that's what we're kind of talking about.
So I'm not. Does someone want to Google this
Yeah.
and just like look it up and then like tell us what it is? Because that would actually help us. Sorry, can you like you can actually like just speak up then so we can like we can benefit.
Oh, it's the simian sarcoma virus.
Okay, so
That's
cool.
cute. Okay.
So probably what we're seeing here is that it's probably coming from a virus, and maybe the I'm thinking maybe like the genetic information of the virus is somehow leading to the overproduction of platelet-derived growth factor, right? Right.
Okay.
I don't think he develops this in a lecture, so like some of this stuff is it's just like
Not even say that.
okay.
I'll buy it. That's all. I'll take it.
Okay. All right. So okay. So that was for the growth factor. This is the growth factor that he mentioned in class. Now for the receptors, all right. So the main ones that are mentioned in class are your tyrosine kinases. Now, when you're looking at those growth factor receptors, right, they're going to be tyrosine kinases, right? And I will show you guys how the receptor works in a couple seconds. But specifically, you have your HER2, right, which is your human epidermal growth factor 2, right? So this is the first growth factor that was mentioned, is a tyrosine kinase. Now the other one, actually let me see. Okay, well the other ones that were mentioned that are also tyrosine kinases is your KIT and your platelet-derived growth factor receptor alpha. Okay. All right. So what are we saying? So basically, these receptors are tyrosine kinases. All right, and because they're tyrosine kinases, they're basically going to be sorry.
Are they types of growth factors?
So they're
Growth
types of
factor receptors?
growth factor receptors, exactly. Okay, so let's actually look at these tyrosine kinases and try to get an idea of how they're actually working. All right, so we're looking at a tyrosine kinase receptor. Okay. Okay, so this is possibly a growth factor, right? And so what's going to happen is that one of these guys is going to bind onto here to one of the subunits of the receptor, and then the other one is going to bind to the other subunit of the receptor. So these guys are going to be like separate, and then once they each of or the growth factor binds to I guess each subunit of the receptor, then they come together and then they dimerize. All right, and that's what we're seeing here. All right, and once they dimerize, all right, the kinase activity is stimulated. All right, so you start getting all these little phosphorylation here. All right, now once that you get all these little phosphorylations going on here, then you have proteins that can just like hook up here, get phosphorylated, and then they can go, and then they can do other things. All right, so this is how your tyrosine kinase receptors are working. All right, so what were the tyrosine kinase receptors that we just mentioned? Okay, so HER2, the KIT, and then what else? Okay, PDGF alpha. Okay. Okay, cool. All right, so this is how it's working. Okay, now when we're specifically looking at the HER2, all right, the HER2 generally leads to breast cancer. This mutation, you generally see it in breast cancer. All right, so basically you would have an overproduction of HER2, right? Because that would make sense. That's if you have a whole bunch of those receptors, then if you had a little bit of growth factors present, it'll be very easy for them to bind onto the cell and then stimulate proliferation, right? Right. So, but HER2, HER2 overproduction, you see it in breast cancer, but it's not specifically like it's not only for breast cancer alone. It can be present in other. It can be a mutation in other cancers, right? So I already said it. So HER2 is going to be overproduced, and because it's overproduced, it's going to lead to increased growth and division. All right. And so how does it lead to increased growth and division? Okay, so number one, it's gonna inhibit apoptosis. So we're gonna see the pathway in a little bit. So, but it's gonna inhibit apoptosis downstream. Some of its downstream effects is that it will inhibit apoptosis, right? So if it inhibits apoptosis, then the cell is left to, you know, proliferate, and it can also increase. It can lead to an increase in cell cycle promotion.
One quick question.
Yes.
So the receptor directly inhibits apoptosis?
The HER2,
Yeah.
the downstream effects,
Okay.
and we'll see that in a little bit. Okay, so here it goes right here. Okay, so here's your HER2, right? So HER2 is what kind of receptor? What kind of receptor is HER2? Tyrosine kinase. Okay, so tyrosine kinase, how does it work? Okay. Okay, so growth factors go on the subunits, they dimerize, cross phosphorylation. Okay, so cool, that's what we're seeing here, right? So we would have some kind of growth factor here binding onto here, right? HER2 receptor. You know, we have the little phosphorylation here. These, you know, whatever this protein is, whatever it is, comes on here. It leads to a bunch of different things going on here. Essentially, this pathway here leads to cell cycle progression. So instead of blocking the cell cycle, we're allowing the cell cycle to continue, so that leads to more divisions, right? Then with this pathway here, all right, I'm going to point out some of the things that I feel like are critical. So that, you know, you don't need to know like every one of these little things, right? So in this pathway here, it promotes, well, it prevents apoptosis, right? So it's anti-apoptotic, all right? Now, I think this is important. So BCL2 is anti-apoptotic. It's an anti-apoptotic protein, all right? So I think that's important for you guys to know.
Anti-apoptotic?
It's anti-apoptotic. So that means that whenever it's around, it's going to prevent apoptosis. So it's going to prevent natural cell death. Bless you. Cool. Okay, so this pathway is also important because we're going to see it again. So I'll just kind of mention it, but we'll see it again.
Quick question, you said anti-apoptotic protein?
Yeah. I mean, of course it has a gene, right? But yeah, that's a protein. Okay, so now this guy, I don't know how important this guy is, but this guy leads to, it will turn RAS GDP to RAS GTP, right? RAS GTP will turn RAF inhibitory to RAF activated. RAF activated acts on MEK, right? And then MEK acts on ERK, right? And then ERK can then enter into the nucleus. We'll see this again, but I just want to just like, I guess just point it out for now. Okay, now we're looking at another example here, and in this example here, we're looking at the KIT signaling pathway. Sorry.
So on the last slide, I'm just
Yeah.
trying to figure out like what is going to cause, because aren't these opposing pathways?
They're not opposing pathways. They're actually leading, both of these are leading to cancer.
Oh, cell cycle progression. Okay.
Yeah, so if the cell cycle is, you know, like if you're in the G0, right, your cell cycle, you're not like, you know, you're not actively going through the cell cycle and dividing, right? But if you're actively going through the cell cycle and dividing, then it's going to promote hyperplasia. Now, if there's something wrong with the cell, you don't want that cell to keep on dividing, right? You want to promote apoptosis so that we can get rid of that abnormal cell, so that we don't have a whole bunch of abnormal cells. So, but when the cell starts to promote anti-apoptosis, meaning it's allowing abnormal cells to like run loose or like divide, then you're gonna get your dysplasia. Hyperplasia because the cell cycle is continuing. Dysplasia because you're allowing abnormal cells to continue. You see what I'm saying? Yeah. Does everyone see this? Okay. Okay, so we mentioned the three growth factor receptors that were mentioned in lecture. So we have the HER2, right? Then we have the what else? We have the KIT, and then what else? Okay. Okay, so then the KIT, what kind of receptor is it? Tyrosine kinase. So how does tyrosine kinase work?
Growth factor binds, dimerizes,
Okay.
cross phosphorylation.
Okay, terrific. Okay, okay, so the KIT, your HER2, the PDGF alpha, they are all tyrosine kinase receptors, right? So for the tyrosine kinase receptors, you have the growth factor binding to the subunits. Then once they bind to the subunits come together, and then you get cross phosphorylation. Then you have other appending proteins or factors that come and they get activated, right? And that's what we're seeing here. So I'm just showing you guys an example with the KIT. So what actually binds to the KIT receptor is your stem cell factor. It binds to your the KIT receptor, right? We get the dimerization by cross phosphorylation. All right. Then you have activation of your RAS, right? So from RAS GDP, right, to RAS GTP. This is important because there's actually a mutation of RAS coming up. So you get RAS GDP going to RAS GTP. Then RAS activates RAF. So RR, right, go together, RAS, RAF. Then MEK, ERK, right? So RAF activates MEK, and then MEK activates ERK, right? Now another way you can remember this, so E is on MEK, right? So then E is ERK. So RAS, RAF, MEK, then the E, ERK, right? That makes sense, right? These are a little bit related. You can see, I'm just trying to give you guys a way to remember this. Sorry, go ahead.
Are we gonna have to know this?
Okay, so from the lecture, this is what, and again, I'm going from the lecture, all right? Where is it? Okay, so here it is. Pathways that you should know. So
Thank you.
it wasn't a lecture. 1 second, let me pull this up. Let me actually pull this up.
Or did you?
1 second, let me pull up the lecture.
You put videos on there.
You did?
Like videos.
Oh yeah.
That's why you skip through.
Okay, so these were so he referenced, so these are signal transductions. This was pointed out, right? The Wnt pathway, right? And this is the one we were just talking about, right? So RAS, RAF. So you have, this is your growth factor. Well, this is the growth factor receptor, right? Then you get the RAS, then the RAF, then the what? The MEK, then the ERK, right? I'm just giving you guys a way to kind of remember some of it. Signal transduction pathway, this is generic, right? And then G protein. But I'm trying to go into the ones that were mentioned in the lecture, right? Because I don't want to give you guys information you don't need to know. Right? Yeah. Okay, so where were we? Okay, so so we talked about HER2. HER2 is common, is going to be mutated where? Breast cancer, right? You can also see it in other cancers. So if someone comes in and you see, well, I guess if you have overproduction of your HER2, it's a poor prognosis, right? For ovarian and breast cancer. So essentially, when this mutation is present, the likelihood that that mass that you have being benign or malignant, so it's going to be more so malignant than it being benign. You see what I'm saying? So if someone has a mutation, or if the lady has a mutation in the HER2, it's going to be more likely that that lump in her breast is more so malignant than it being benign.
Wait, in lecture today, I asked him this question. He said, he was talking about the HER2 positive is only present in 20% of breast cancers.
20%.
Most of them, yeah, most of them aren't HER2 positive. Then he mentioned about triple negative and
Yeah, that's other
Okay.
Other
okay.
pathways that
Right.
cause breast cancer.
Yeah.
That's,
But we're just talking
Yeah.
about the ones
you just
specific.
talk about one. So most of the time in
It-
cancers, you're talking about different pathways that are going wrong.
exactly. That's right.
And
Yeah.
involving the HER2.
Yeah, HER2
Yeah,
is
HER2
one of many pathways.
Okay,
yeah, it's
okay,
one
just
of
meaning
many.
it's involved, not
Yeah,
just the
Only
exactly.
way, not the
Yeah,
only way.
yeah. It's not
Okay,
the only.
okay.
Yeah, it's not the only way. Remember what we said, right? So when you're looking at that malignant tumor, is a series of mutations that
Right.
cause that malignancy. So HER2 is just one of several things that has gone wrong. But it's just that when you have breast cancer and it's malignant, you can suspect that HER2 mutation is present. I mean, like, like it's more likely for that mutation to be present. I don't know if that makes sense. Okay. Yes, maybe. Okay. Okay, so we saw that HER2 is correlated with breast cancer. Now, when you're looking at KIT, KIT and your PDGF receptor alpha, so they are associated with hereditary gastrointestinal stromal tumors. Hereditary gastrointestinal stromal tumors. So just
So basically, that's
Can you say that?
okay. So if you have a mutation in your KIT or your PDGF alpha, so it's associated with hereditary gastrointestinal stromal tumors. So the same way that there's a correlation with HER2 mutation in breast cancer, it's the same way that you can correlate KIT and PDGF receptor alpha mutations with hereditary gastrointestinal stromal tumors. So it's kind of like a correlation kind of a thing, right? Did everyone get this? Yes?
Yeah.
Okay, so we looked at what? We looked at the growth factor. We looked at the receptor. Now we're looking at the G protein. All right. So remember, it was what was it again? It was RAS. One second. One second. Let me make sure I get this. Okay, so it was what? RAS, RAF, right? Then it was MEC, and then it was ERK, right? So you can have a mutation with that RAS, all right? So if that RAS becomes mutated, so that means it's oncogenic, right? And we can see that with the KRAS. Okay, so we can have a mutation in the RAS, and that can lead to, or it can promote the cancerous state. And then we could also have mutations with the nuclear transcription factors, right? And again, I'm specifically giving you guys the ones that were mentioned in class, all right? You can have a mutation with the MYC, all right? This is a nuclear transcription factor, all right? If you have too much MYC, or if you have an overproduction of MYC, or if you have an overstimulation of MYC, it's associated with cancer, right? And let me see if I can get you guys an example here. Okay, so if you see here, right? So with the HER2, right, you have your growth factor binding to the receptor, right? It dimerizes, cross phosphorylation. This protein comes on, gets activated. It turns RAS GDP to RAS GTP. All right. Now, we talked about the RAS mutation. When the RAS is abnormally, whenever the RAS is abnormally in its phosphorylated state, that's when it's cancerous. All right. Whenever RAS is in its phosphorylated state, meaning RAS GTP, that's when it becomes cancerous. All right, because then it can act separate from all this, right? it can just act by itself. It doesn't need any stimulation from the growth factor, right? It can just act by itself and then lead to the activation of the rest of this. All right, so we got the RAS, RAF, MEC, ERK, right? Then the ERK goes in and it leads to the transcription of now your CMYC. Now we just said that the MYC was the transcription factor. So if this MYC transcription factor, if it's for some reason overstimulated, or if too much of it is produced, it can lead to cell cycle progression, which can lead to abnormal division, which can promote the cancerous state. All right, so did everyone get that? Cool? Yeah? Maybe? All right, so I'm going to ask you guys a question. 1 second here. Yeah, go ahead.
We didn't really go through the second side of this kid signaling pathway.
Yeah.
Is that like
Important.
I don't think it's critical.
because it's for survival?
Yeah. I guess the fact that it promotes survival is important, but all this is not in any of those pathways that I saw in lecture. So we just kind of block this out of the way. This doesn't exist, at least for now. Okay. Let me just ask you guys some questions, and you guys feel free. I'll just put the questions here, and then you guys can just like discuss with the person next to you, and you guys can try to come up with the answer. So you don't have to tell me the answer. Just talk with the person next to you. Alright, so just talk to someone next to you, and then you guys can try to come up with the answer.
They're so humid. I feel like
I'm sorry,
I can
man.
slice it.
I'm sorry, man. I think it probably has like a limit number. It has a number.
It could be like you can't publish.
Alright, so did you guys get this one? You guys got this one, right?
Yeah.
Okay.
That's exactly what it That's exactly what it increase like
Well,
reduction of receptors.
that's like the receptor itself.
Oh, is that the ERK?
Okay, yeah, that pathway. What does it lead to at the end of the day?
Ultimately it leads to anti-apoptosis and like
Okay.
continuation of cell.
So you're speaking to me, right?
Yeah, well,
As you run.
oh, what?
I'm just, mean, I think it'd be better to speak to her. You know,
Okay.
you're right,
So,
scared.
You get 10, right? Dude, they're probably just off.
No, it's also like self-preservation, like a continuation. Like one way you can go, you can actually just solution, and the other way
Yeah. I would imagine. I don't think it's enough.
yeah, you like you have one way that makes but then the last half,
You've seen how long
into
it's
that self may you have the C and like yeah, it's leads to increase the make cells and then they don't want to die. Yeah, the growth factor.
The cascade factor, growth receptor, and transcriptase factor.
The PDGF, and then you have the and then you have the
So pretty much everything that promotes sexual activity.
Exactly. Exactly. Okay, so maybe just one last question, and then we can just jump back into this.
See, I'm thinking like that's what you're talking about. Oh, that's what you think. But it's like break it. So do those always result in cancer, or is it? Oh, that's what you're talking about. The steps to mutation. That's drawing blood supply. Or you mean like invading other tissues, drawing
Yeah,
blood
that too.
supply, and then entering the blood vessel. Yeah, like a progression of mutations, like the result of mutations, not the
Okay, so you guys good?
Okay.
Did anyone have any questions? Sorry, did anyone have any? Yeah, sure. Go ahead. This is being recorded, so oh, I'm sorry. I'm sorry.
These are acquired because they have inherent mutations over time, so they're not inherited genes.
Okay, so it's a series of mutations that occur. Now, so again, it's kind of like a cascade. So like the fact that you have more proliferation of cells means that there is a higher chance of you getting a mutation. So like maybe like okay, so there are cancers that are, that I guess that can be inherited, or genetic mutations that can be inherited, but I guess so over time you are getting these mutations that are occurring, but I guess the way that they can be coming about is that you're getting a rapid proliferation of cells, which leads to gives you a higher likelihood of having more mutations because of you get all this cell division, and then what I'm saying, then you can get more mutations because of that. I don't know if that kind of makes
It's
sense.
just like multiple factors that lead to like your lifestyle, like diet, smoking, stuff like that can lead to mutations that lead to hyperplasia dysplasia, or like,
That's all.
damage, like cutting yourself, that can lead to inflammation. If you don't take care of it, it can lead to cell proliferation and mutations and hyperplasia dysplasia, etc.
So like external effects.
Yeah. So not genetics.
Yeah,
Not genetics.
Yeah.
Yeah.
Alright, cool.
So I wouldn't just so you can't just only like circumscribe it to genetics or external factors. So there's multiple things that I'm saying. It can be genetic, it can be external. It can be the food, carcinogens, so many different things.
Environmental.
Exactly. Yeah.
Can I ask a question?
Yeah.
So the drawing of blood supply,
Yeah.
wouldn't that already make it cancerous? If it's just like doing something and it's like making blood vessels.
Well, I guess. So like actually angiogenesis. So like you're actually getting blood vessels feeding into that cancer itself. You know what I'm saying? Not that the cells are not like using a lot of your body's nutrients, but actually they produce factors that allows the I guess blood vessels to grow towards where the cancer is.
Yeah.
You know what I'm saying?
Right here to the basement membrane.
Yeah.
Is that like aberrant activity? Is that like something random, like an accident? Like
Okay, so essentially, so for the cancer to be malignant, all right, it has to have metastatic potential. So it has to have metastatic potential. Now, if you're going to metastasize, now you're not going to be in the same location, right?
yeah.
So you're going to have to move around. And so the first thing you have to do in order to move around is you're going to have to remove that restriction, or you're going to have to break away from that basement membrane. All right, and so you break through the basement membrane, you invade other tissues, maybe the fibroblasts, then you get into the blood vessel. From the blood vessel, you can now, you can move around the body, and then now you can go to a different location. All right. Okay, cool. All right, so we talked about the proto-oncogenes. So really fast, just going with the tumor suppressor genes. So tumor suppressor genes, they're going to prevent the cell from dividing, right? And is also going to lead to like apoptosis and stuff like that. But these ones were the ones that were specifically mentioned in class, so I'll just point these ones out. So you have a cell cycle regulator, P53, all right? You have a DNA repair gene, so you're a BRCA1. And this one is a little bit different, so you beta-catenin and your E-cadherin. And I'm going to go into these in a little bit more detail, but I think I'll point them out first before we go into them. So the beta-catenin and the E-cadherin, they're related to APC. We're going to see that in a little bit. Okay, so going with this P53, so essentially when you have damage to your chromosome, the P53 can detect that, and it can lead to the apoptosis of that cell, because you don't want that cell to continue to divide, right? That will create a problem, right? So P53 will promote apoptosis, so we can eliminate that cell with abnormal chromosomes, right? Now, another thing that P53 can do for you is that it can arrest the cell cycle. So once it notices that, oh, you know, there's something wrong with the chromosomes or the genetic material, it can arrest the cell cycle and then allow for DNA repair to occur before that cell starts to divide again. And that way it can be suppressing the tumor, or that way it's functioning as a tumor suppressor. Does this make sense? Okay, so that's what your P53 is doing for you, right? Now, when you have problems with your P53, all right, now you're going to get, it's going to promote what? The cancerous state, right? Now, a condition where you have a mutation of your P53 is your Lee-Fraumeni syndrome. So this is where you have a mutation in that P53, right? Okay, now your BRCA1 is going to be promoting a DNA repair, right? So you need if there's something wrong with the genetic information, you need it to be repaired so that everything in that cell can be normal, because what? If the DNA is messed up, protein's going to be messed up. So the BRCA1 is promoting DNA repair, which is essential for having normal, right, cells. All right. Now your beta-catenin and E-cadherin, I'm sure you guys have an idea of what E-cadherin is already.
Not a
IgG.
Hmm?
You said IgG.
Not exactly.
Cadherin like
Cadherins are important for cell-to-cell linkages and cell-to-cell communication.
Exactly. All right. Okay. Yeah. So that's exactly right.
Can you repeat that? Because I don't know.
We'll see it in a little bit. We'll see it in a little bit. And you guys are so anxious, like. Okay, so here we have a receptor. So in the slide, it did talk about knowing this Wnt pathway.
Whoa, what?
This Wnt pathway.
I don't know what that is.
I'm not sure what Wnt stands for, but you should know the pathway or have an idea.
How are you going to teach
Have an idea how that. Okay, so there's a lot of confusing things here. All right, so for now, just ignore this.
All right. Cut that out.
And then ignore this as well.
Nothing. Now we're working with
Okay. Actually, let me start with something a little bit easier.
Yes.
You'll get it in a little bit. Just have some patience.
Work it out.
Okay, so you have Wnt. So I'm supposing it's some kind of Protein or ligand, perfect. And it binds to the frizzled receptor, which is going to lead to, I guess, activation of disheveled, which is going to act on GSK3 beta, which acts on APC. All right, so remember we just saw APC, right? Let me see APC. So APC right here, right? That APC is related to your beta catenin and E-cadherin. But I'm showing you, so that the APC is with this Wnt pathway, right? And that APC then, I guess, allows for beta catenin to be released, and this can enter into your nucleus and cause gene regulation. But specifically, it can also do other things, and that's actually what we're interested in. So this is just another diagram here. Okay, so the Wnt binds to the frizzled receptor. I believe this should be the disheveled.
Yeah.
So the disheveled would inhibit GSK3 beta, which will, I guess, then release this from inactivation.
APC?
Yeah, I believe. That's what I believe. I could be wrong. And I don't think you're supposed to know it in this detail, but I'm just trying to make sense of this. All right. Then the APC will lead to beta catenin stability, or I guess more beta catenin. This beta catenin can now work in tandem with your E-cadherin to form this adhering junction. And the adhering junction is what keeps the cells together. Okay, so for you to have a malignant cancer, that cancer has to be able to metastasize. That means it has to be able to move from one place to another place. Now, if that cell is linked to all the other cells, how's it going to metastasize? Right? So we have to what? We have to remove this adhering junction, essentially. So we need a mutation in this APC, essentially. If we have a mutation in the APC, right, then we can't form these adhering junctions, right? Now the cell can, move somewhere else because it no longer needs to be connected to anything else. Does that make sense?
So it keeps cells together.
Exactly.
So if you have a mutation,
Yeah, then they can. Does this make sense?
yes.
Okay, cool. So this is for your tumor suppressor genes. So remember we talked about the proto-oncogenes. This is for your tumor suppressor genes. All right?
So yeah, if you have a mutation there, the cancer will spread to other places.
Yeah, for the beta catenin and the E-cadherin.
Wait, I have a question.
Yeah, go ahead.
Back to the one that you draw. So it says disheveled inhibits
I think GSK3 is I think it's normally holding all these maybe like in an inactive position.
So then the communication to APC that blocks
It's the mutation in the APC will then prevent, I guess maybe prevent it from acting on the beta catenin.
Okay.
You see what I'm saying?
Yeah, so why is there inhibition thing there?
So I guess that the GSK3 beta normally holds the APC and something else here in an inactive position, so that when the GSK3 is inhibited by disheveled, whatever, then it can release the APC to be in its active form or whatever. That's
why the normal doctor.
Can you, real fast, I'm sorry.
yeah.
just
No,
real
you're
quick,
fine, dude.
Can
You're
you
fine.
like erase like the red on the side?
Yeah, sure.
So
Yeah, whatever you want, man.
Okay, that's cool.
All right.
Thanks.
Yep. Are you good?
Yeah, okay. Thank you.
All right.
So APC when it's phosphorylated, it's
Okay, so we're talking about cancer, how we start out with a normal cell, it gets all these mutations, and then it becomes a cancer. So we talked about what is getting mutated, all right? But we're not actually talking about the forms of the mutation, or like how that mutation is actually occurring in the, I guess, like the genetic level, all right? So we're seeing that certain things are getting mutated, all right? But the question is, how are they getting mutated, all right? So you can have a point mutation or deletion, all right? So again, we're looking at the chromosomal level. We saw that you can have mutation in your proto-oncogenes. Then we saw that you can have mutation in your the tumor suppressor genes. But on the chromosomal level, what is actually occurring, or what can happen? So you can have a point mutation, all right? This is how we're getting that abnormal RAS, all right? So we saw, where did we see RAS? Where did we see it? Huh? Okay, so growth factor. So from RAS, we have what?
RAF.
Then we have RAF. Then from RAF we have what?
MEC.
Then MEC. Then we have what? Okay, good. Okay, so we can have either a deletion or point mutation, right? And the one that was specifically pointed out in lecture is this RAS, right? So or we can have a gene amplification, right? So you can have, I guess, more segments of that gene, right? So gene amplification, you have more segments of that gene leading to an overproduction of the protein product. This is what we see with MYC. Now, what was MYC again? What is MYC? The transcription factor. Okay, so transcription factor. So is this a tumor suppressor gene, or is it a proto-oncogene? Okay, so what is a proto-oncogene? Okay, cool. Okay, so we can get point mutation, gene amplification, or we can get a chromosomal rearrangement. All right. Okay, so the essential idea with this chromosomal rearrangement is that you're taking genes, or you're taking a genetic information that is not readily produced, and you're taking that information and you're putting it next to some genetic information that is normally like constitutively produced, like it's always being produced. All right. So the two cases that were pointed out is with the chronic myeloid leukemia and with Burkitt's lymphoma. All right. So when we're talking about this chromosomal rearrangement, we're saying that there is a gene, right, and that it's not readily transcribed. So we have a certain amount of the protein product. But what happens is that when you have the chromosomal arrangement, we're taking that genetic information and we're putting it somewhere or next to a gene that is actively being transcribed. So what is the net effect? The net effect is that that gene that wasn't normally being transcribed a lot is now being like actively transcribed. Does that make sense? Okay, so when we look at this chronic myeloid leukemia, so it's going to be a translocation of between chromosome 9 and 22. All right. So here you have. I'm just looking at time. I don't want to hold you guys here too long. Okay, so normally this is chromosome 9, all right, and this is chromosome 22. Now this is your B cell receptor. So the B cell receptor is actively being like produced. It's actively being transcribed, right? So what happens is this piece here, ABL, whatever, can now be removed, chromosomally removed, and this piece here can be removed, and they can exchange positions. And so now this ABL is next to this BCR. So this ABL can now be, it's being transcribed much more now than it was before. So we have a lot more of that protein product. Does that make sense? Yeah.
Is that just from like cross-linking, or I mean, is it just like, I guess, that's a pop off and
I believe this is genetic from now on. I think this one is genetic. Chronic myeloid leukemia.
So what would cause the chromosomal
I'm not 100% sure. Someone Google it, and then maybe you guys can just like fill us. Okay, so but it leads to production of active tyrosine kinase. So I'm thinking maybe it's leading to overproduction of your tyrosine kinase receptors, right? And because you're having a lot of these receptors, then that can promote the cancerous state. That makes sense. Cool. And then when you have your Burkitt's lymphoma, so again, it's the same concept, right? So you're having something that is not actively being transcribed, and it's moved to a location that is being actively transcribed, and you can see it here. So your c-MYC, right? So it's normally on chromosome 8, and in this position, it's not being actively transcribed. But however, the genetic information for your IGH is. So when you have this chromosomal rearrangement, right? Now this c-MYC is next to this IGH. So now this MYC, which we said was a transcription factor, can now be produced in larger quantities, and it will act as a transcription factor, right? And then it will promote, right, the cancerous state, right? Does this make sense?
Yeah.
Okay, so on the chromosomal level, what we're seeing is that in terms of the mutations, we can have point mutations, all right? We can have gene amplification, all right? Now, I know I didn't point this out. So when you're talking about gene amplification, it means that instead of having one copy of that genetic information, you're actually having multiple copies of that genetic information, all right? Does that make sense? Or you can have a chromosomal rearrangement, which we see here. Right? Cool. One second, I'm gonna ask you. So I don't want to hold you guys here too long, but I don't want to just keep talking. So I will ask you guys some questions, and you guys can just speak to the person next to you.
Is that the main stuff that is that everything that he wanted us to know as far as you're concerned?
Okay, so for the most part, I put everything that was on the slides on here. Maybe just a few minor details that I didn't put on here, but everything from that lecture is here. Everything from that lecture is here, yeah.
Is there anything,
Yeah.
sorry, is there anything on the lecture or that you could grasp that would correlate to the title?
Yeah, so that's what I was saying. So the title of the lecture is a little bit misleading, but essentially, in the lecture, he just talks about cancer and how how the cancer, like how it comes about, what genes are mutated, and then later on, as he develops the lecture, he goes into like specifically colorectal cancer. Yeah, that's essentially what the lecture. But it's a little bit misleading. A little bit misleading. Yeah.
He has like a single slide or something about this.
Sorry, I didn't hear you.
Colorectal.
Wait, so are you talking about the drugs?
Yeah.
Okay, so I have that on here. So for the most part, I have everything. Again, it's just a few minor details, that I kind of maybe like brushed over because I thought they weren't really all that important. But everything on that lecture is here. Okay, so I'll ask you guys a couple questions here. the gene will be transcribed.
I have a question.
Transcribe a lot more than usual, so you'll have a lot more of the protein product.
I'm barely getting it.
It's an
He's my friend. He's like,
I thought that's what they do it regularly, but when they're that's what they do regularly, but then when they're mutated, they don't. That's what I'm saying. When this is not mutated, that's what it does. And that's the problem. When it is mutated, it's like it does not evolve.
I don't know about his structure of sugars actually.
It doesn't.
Yeah, no problem. You're fine.
Good legs.
Shut down. So then either have DNA damage and stay not real or you have decreasing in calcium. That's what happened mainly with like the 3, but then for the rest of the one, divided by less.
Wow. So that's why I'm not gonna do it.
Y'all staying or leaving? Here?
Sorry?
Are you gonna go over the little details that you did yet, or is Medstar?
Okay, so all right, so I basically I'm basically like, I'm still like 75% done. The only other thing that's left is to talk about colorectal cancer, and then there's a little bit. He develops a little bit of thing on the treatments, but as you see, everything that I talked about and established in the beginning will help you to perfectly understand everything. Everything should make.
I have no issue staying for the last 25%.
Yeah. So.
Do you want to stay for the last 25%?
I mean.
Not really. That's
No. I mean, okay.
If you guys work it or
Huh?
online on Facebook.
Sorry.
I know he is on Facebook, so it won't.
No, but the actual slides, I think.
Oh, then he's done.
Yeah, he made it, right?
Yeah.
It's on there. We just gotta read it on our own.
Yeah. Okay. So I mean, like, I don't have any problem. I mean, like, I made it so I can go over it with you guys. I didn't make it so I can complete half and then like run away. So.
You're cool. That's what you're saying.
Yeah.
At least it's not 9:30.
Oh, yeah.
Okay.
Yo, that joke was helpful though.
It was.
I didn't
You were
expect
there
to be all that
until
day, and I was
9:30.
That's real. He went over when I that black opera, I knew.
Yeah. It was too late though.
That wasn't his fault.
Okay.
That was
So
his fault.
let's see.
That was nice.
I was good for the next video. I'm ready for the quiz. Yeah.
No.
It was very helpful. Yesterday was helpful too, actually.
Which one?
I got it.
Yesterday was helpful because he went over everything and he broke it down that it was easy to understand.
And he went through the actual PowerPoint, so it was
Yeah, exactly. So like, okay, this is what you need to know, all that.
True, but by writing it, you will
Shout out to the fact that temperature went down by like 10 degrees.
All right.
Okay, what genetic abnormalities lead to cancer formation? Genetic? Oh, okay, so that's the,
On like the chromosomal level.
yeah, the mutations,
Like the-
mutations and everything.
Oh,
Yeah.
Okay.
With the myeloid, chronic myeloid leukemia, that's the 9 to 22 translocation.
Right,
And
so translocation is point mutation.
Oh,
Yeah,
deletion.
what was that?
Spelling is
Point,
gonna be harder, but it's
the,
SACHIN.
and that point mutation dealt with RAS, how
Yeah.
like it makes it meets the more active form of the protein, so you make more of that protein, and then it goes through the process. Yeah, Nick was gene amplification, so because you lead to a reduction of a protein. Yeah, and then chromosomal rearrangements where you either have a deletion.
Can you say it?
You have translocations with that one.
I can't get that middle part.
Say it again, say it again.
Huh?
What are we saying?
It's so beautiful.
Don't worry.
What are we saying? I heard French. I wanna hear it.
You see what happens?
Try to play this game, knowing that I don't know where the last words.
Tells everyone the
Point mutation, gene amplification, chromosomal rearrangement. It's a little.
Alright, shut up. I'm gonna be like
All right. So, can you guys give an example of this, of a point mutation? Okay. Okay. So the RAS. You guys cheating?
A little bit, but
I just have a good teacher.
that too.
Do I get brownie points?
I don't have any brownies though.
Can you make some?
I don't know how to cook. Okay. For the gene, how about for the gene amplification? Okay. And then Mick was what? What is Mick? Transcription function. Okay. Perfect. And then for the chromosomal rearrangement, what were the examples?
Yeah, the
Chronic myeloid leukemia
Okay.
and Burkitt's lymphoma.
and Philadelphia.
Okay. Chronic myeloid. So what type of translocation is the chronic myeloid leukemia?
9 to 22
Okay.
translocation.
922. That's right. Then the other one was
Burkitt's lymphoma.
Burkitt's. Okay. I kind of have a crazy way of remembering this. I don't know if you guys want me to share this with you.
Sure. Yes.
Okay, so this. Okay, so the B looks like an 8, right? So that can help you with the 8. And then the lymph. So I mean, like it's lymph. Like I think of like 4. For some reason, I think it's lymph. Yeah, lymph. Yeah. It's good. Lymph. Yeah, lymph. Yeah, lymph. Yeah. So
The L and
lymph.
the Y kind
Lymph.
of look like a 4 the way you draw it.
Yeah,
Okay.
the Y does look. That does look 14 actually.
I'm trying to get you guys, maybe give you a way of remembering this example. So lymphoma, 4 phoma.
Oh, from the
And
L.
then
The L can be the one. So you have the
okay, okay, that's even better. That's even better. That's even better. That's even. Okay, so lymph.
Something's gonna stick. More of the story is something.
Right. So if it's not this, then it's that.
Right.
That's a fact though.
And that is chromosome.
Chromosomal
Rearrangement.
Okay, so we're ready to move into the colorectal cancer. Are you guys situated?
Now that all the people done left, it's a lot cooler.
All right, so okay, so just a little bit of background information about colorectal cancer. So the colorectal cancer is the 3rd leading cause of cancer by both incidence and mortality. So in terms of like, when you look at cancer and you look at what is the 3rd leading cause of death associated with cancer, it's colorectal cancer. When you're looking at like how many, like what cancer like is frequently gotten by people or like affecting people, so the 3rd highest would be colorectal cancer. So it's the 3rd leading cause of cancer by incidence, meaning by occurrence, and then also by mortality, like how many people die from it. Cool. No, it's not the first. The mortality. Let's see. So mortality, I believe it should be. I don't think it's cardiovascular. No, it's not prostate. It's lung cancer. Lung cancer is first by mortality, then prostate and breast cancer is first by incidence. Okay, so a little bit on the timeline. Okay, so normally, like if you start off with your normal intestinal epithelium, all right. Now, so you normally, when you start to get all these mutations and stuff like that, you will transition to your adenoma, or like the polyps, which are benign. And then as you know, you get more mutations, right? That's when you get the carcinoma. So the adenoma is benign in the sense that it's not metastatic, so it doesn't have metastatic potential.
But it is cancerous?
It's precancerous, so, but it's not really deadly. Okay, so this is what I got from the slide. So this tumor progression takes about 10 to 35 years to reach malignancy. So generally. Okay, so what are we seeing here? So we're seeing the normal colon epithelium here. Right? This is from lecture. So this kind of looks bad, Okay, so this is the normal colon epithelium, and now this is some crazy stuff. All right. So you have okay, so you have all these polyps that are in the colon, right? You have these multiple polyps. Okay, so now there's kind of like a sequence to this, right? All right. So generally, the first mutation that occurs generally is a mutation of your APC gene. Okay, now backtrack. Where do we see APC gene?
Tumor repressor.
Tumor suppressor. That's right. Related to what? Tumor
Adherence.
Cadherin. That's right. Cell contact. That's right. So, okay, adhering junction. So the first thing that's going to happen is that these cells are going to lose contact with the neighboring cells. It's the first thing that happens. All right. Then, in terms of like the mutations, how the mutations are coming, then you get a mutation in your KRAS. All right, and that was it's a proto-oncogene, right? We established that. So from RAS, you have what? RAF, right? MEK, ERK, right? Then that can have effects, right? Then you get a mutation in your DCC. Now we didn't talk about this. We didn't talk about DCC, but just know that it is a tumor suppressor gene. If someone wants to look it up for us and just give us just a little bit of just a filler on what DCC is, that will be appreciated. Okay, so we have DCC. Then you get a mutation in your P53. All right, so where do we see P53?
Oh, that's tumor suppressor.
Tumor suppressor. So what did the P53 do for us?
Apoptosis, and it can end the cell cycle.
Okay, exactly. So normally, if you have some kind of chromosomal, problem or whatever, the P53 can detect that, and then it can halt the cell cycle until that problem is corrected, or it can just send for apoptosis of that cell with that problem, right? Cool. So these are like the sequence of mutations that are occurring, all right? And yet you get other mutations that occurs that leads to this colorectal cancer or this carcinoma.
So DCC is actually called deleted in colorectal carcinoma. It's called deleted in colorectal carcinoma. That is the full name of DCC.
Deleted in...
Self-explanatory.
This is so dumb.
Yeah.
Why would they name this? Like, why would they...
No idea.
I guess they ran out of names. I just ran out of.
That's why they probably keep it.
This is so dumb.
So it sounds.
The figure is meant to school is too high.
Right.
It should be called by the first name, like Bob's. Albert.
Okay, so deleted in colorectal cancer. Okay, did you see anything else about it?
Read that shit right now.
It says encoded by the DCC gene, long been implicated in colorectal cancer. Product. The protein product of DCC is a single transmembrane receptor, also known as DCC. It has the same interchangeable name.
Okay.
Axon guidance receptor that responds to netrin 1.
Okay, it's all good. Just know DCC is part of the mutations that leads to the generation of this colorectal cancer. Again, he doesn't develop this in lecture. I'm just trying to fill you guys in so that this information actually makes sense, because it's so much easier to remember things that make sense. I'm serious. I'm very serious about that. If it makes sense, it sticks with you. It's a lot easier. Right. So like now, if you see DCC, it will make a little bit more sense, because now you know it means deleted in colorectal cancer. Right. Okay. Let's see here. Is there anything specific I want to point out here? Okay. Now, so he talks about the progression of the mutations. There's a particular slide I want to show you guys really fast. And again, like everything, if you look through this, like we covered all this. Cancers. Again, like this is just telling you by incidence. So this here you can see the colon rectum. So you can see like it's third, right? By incidence and by death is third. You see that? Right. This is just saying the same exact thing. Like by incidence is third. By death is third. That's all this is saying. Right? See, this is the 10 or 15 years we just talked about, the same picture, right? So this is normal, and then this is bad. Okay, this is what I wanted to point out here. Okay, so if you start off with your normal colon, right? So the first mutation that occurs is the loss of APC tumor suppressor gene on chromosome 5. I know you kind of see, you start to get a little small polyp growth, right? Then you get this benign precancerous tumor growth going. Then you get class 2 adenomas, benign. And again, you're still getting these mutations occurring. Now the next mutation in line is the KRAS, right? Right. Then you have your DCC. Then you have P53. And then you have other changes that eventually leads to this nonsense here. Right? You guys see that? I'm just pointing this out.
Loss of DCC, so loss of deleted in colorectal carcinoma.
Exactly. Perfect. You guys are catching on. Okay, cool. Okay, so now we're getting into the treatments of the colorectal cancer. Right? So it turns out, and again, like there are other mutations that can occur apart from the ones that we've mentioned already. Right? So it turns out that there can be a mutation with your epidermal growth factor receptor. Right? So in addition to all these mutations here, there can be, right, other mutations, right, and specifically with your epidermal growth factor receptor mutation. Now, if we're talking about growth factor receptor, well, what is that? Is that a tumor suppressor or is that a proto-oncogene? Proto-oncogene, right? So now if we have too much of that receptor being made, then that can promote cell division, cell growth, and then that's cancerous, right? So if you have a mutation with your epidermal growth factor receptor, Now, this can this is one of several factors that can lead to the colorectal cancer because the cancer is an accumulation of mutations. But if you have a mutation in this epidermal growth factor receptor, all right, one way that we can treat this is to target it with cetuximab and panitumumab. All right, it's a crazy name. They're pulling this stuff out of the bag. I don't know where they're pulling it out from, but they're pulling it out from somewhere. Okay, so cetuximab and panitumumab. I'm probably saying this wrong. Okay. Okay, so it's acting on the epidermal growth factor receptor, right? So these drugs, they're acting as monoclonal antibodies. So antibodies are binding onto your proteins. All right, so that receptor is a protein, and that drug is acting as an antibody binding onto the receptor, preventing the ligand from coming in. So if we have too much of that receptor present, well, we can just block it. If we block that receptor, then the epidermal growth factor can't bind to it, thus it can't stimulate, cell division, right? Does everyone see that progression?
So I guess it's more so
Blocks
like,
the receptor.
Yeah, go
I
ahead.
guess it's more so like a competitive inhibitor then.
Let's see.
With that
Yeah, I guess you can see it that way. Yeah, because if it's competitive, it'll have to be at the same site. Yeah. So yeah, you're right. You're a smart guy, man. You're a smart guy. Okay, so this is just another figure just to emphasize the point. So here we see that receptor, epidermal growth factor receptor. And again, remember, we're talking about this in the context of colorectal cancer, right? So we have mutation in the epidermal growth factor receptor. Now, the mutation is resulting in the overproduction of these receptors. So what we can do is we can have these monoclonal antibodies. Oh dang, this thing stopped writing. 1 second here. Okay. 1 second here. You guys give me 1 second. All right, so I think we're back on track. Okay, so we have this receptor, and we have these monoclonal antibodies. So what these guys are going to do is that they're going to, bind onto these, supposing that's an antibody, and it will prevent this epidermal growth factor from binding onto this and actually stimulating it. So by binding onto it, then it essentially knocks out the receptor, and the receptor is not able to stimulate the downstream stuff to lead to proliferation, right? Cool. So that's how panitumumab is working, and cetuximab. That's how they're working. So go ahead.
I don't know if you know this, but let's say it binds the receptor, stops it from doing the cascade.
Yeah.
So is the growth factor just hanging around on that extracellular in the plasma, just waiting to bind, or is it destroyed by something? Because you can block it, but it's gonna still build around more.
I think even if it's there, but if it's not active, it shouldn't really have an effect. Because the only time you have an effect is when, really have an effect is when it's binding to the receptor.
Exactly. Yeah. Yeah.
So would it keep building up if it's
Is it
would
if
it
it's too
stop
much
the
and then
production?
negative feedback inhibition where just stop production of it?
Of the growth factor or the receptor?
Yeah, the growth factor. If it's not going, if it's not being transported into
Well, I guess depending on the condition. So if you have injury or something like that, you'll probably want to produce growth factors because you want the cells to actually multiply and divide.
Yeah.
But I don't think there's any negative feedback or stuff occurring. I'm not sure how to.
I kind of get what you're saying.
Yeah.
Okay. And this example here, the mutation is in the growth factor receptor, right? Or is it in the growth factor?
Okay, so the mutation is that we're getting overproduction of the growth factor receptor.
Okay, okay, so
Yeah.
it's okay.
Yeah. There's too much of the receptors
Right, so
around.
it's gonna be a lot more sensitive to whatever little amount of growth factors out there to stimulate.
Exactly. Okay, so is everyone good on this so far? Okay, so we can have a mutation in the epidermal growth factor, or we can have a mutation in that KRAS. Alright? So this is that KRAS right here. So it's either we have too much of the receptor, or we actually have a mutation in the KRAS. Now, typically when you have a mutation in that the RAS, so the RAS is held in an active form. So it's held in the RAS GTP form, which is active. So for some reason, KRAS is in its active form, and thereby it can act autonomously. So it does not need to be prodded by the epidermal growth factor receptor to be active. It's already active. It's its own boss. You know what I mean? So, and it can just lead to proliferation by itself, right? So just some information. So 96% of these mutations, of these KRAS mutations, are going to be occurring within codon 12 and 13. Right? 35 to 43% of colorectal cancer has KRAS mutation.
So the drugs won't be.
Those drugs won't be effective.
Right.
That's right. So just like what's being said, so like cetuximab and pembrolizumab. Okay.
No.
Go ahead. I want to hear you.
No, I can't.
No, but go ahead. I want to actually hear.
Panitumumab. There we go. Panitumumab.
No, but you forgot the other M though. You forgot the first M. So you're just saying the same thing I'm saying now.
That's how you say it, right?
I was actually practicing all this for our game. So just letting you guys know. But so cetuximab and panitumumab, they are not effective against the KRAS mutation because the KRAS is autonomous. It's its own boss. It doesn't need any prodding. It's its own boss. It's already active. Why? The fact that the receptor, if the receptor is active, that's good. If it's not active, it's still active. I mean, like, it doesn't need any assistance. So essentially, if you use panitumumab or cetuximab to treat a KRAS mutation, it won't work because panitumumab and cetuximab are acting on that epidermal growth factor receptor, not on the KRAS.
I have a question.
Yeah.
So these drugs, would they be used when the patient already has cancer or to prevent the progression and ultimate malignancy?
Okay, so we are going to talk a little bit about this. So there is, so this whole concept of molecular profiling. So again, we already established that when you have a cancer, it's as a result of a bunch of mutations. Now, when you want to actually treat the person with the cancer, it will be good to get an idea of their genomic information to know exactly what is mutated so that you can tailor the treatment to that specific person, because you would want to be giving them this panitumumab and cetuximab if they had a KRAS mutation. So it's critical that you know what is mutated so that you can effectively treat them.
Precise and personal.
Exactly, that's right. So that's going with the title. So that's kind of, but I still think it's a bad
Yeah,
it's not.
It's horrible.
Yeah.
But so it's only given, not in the sense that it wouldn't. Okay, it would only be given to the patients who already have cancer, which is precise to their mutation, or it would be given to patients who also have the polyps that are not cancerous yet.
So generally, so if they have this mutation of this epidermal growth factor receptor, give them the drug.
Okay.
So yeah, that's That's the way I see it here. Because if you give them the drug and they don't have that mutation, then it wouldn't make sense.
I mean, but since this is such a slow growing cancer, most people don't detect it until it's too late.
But what if you detect it early? Would you give them the
But
drug then?
but how would you know to check?
Yeah, she's
Proactive.
Yeah.
Oh, I'm
That's
gonna
what-
get a
no,
colonoscopy and see what's going on at age, I don't know,
Yeah,
30
I mean, that's
or
why it's
something.
usually recommended for like 40 and above and all the other crap, something.
When it's too late.
10 years in.
All right, you guys good?
Yep.
Right.
Okay, so now another mutation that we can treat here is the wild type BARF mutation. It's kind of dumb, BARF. Okay.
BRAF.
Okay, BRAF. Okay, so BRAF is seen in less cases of colorectal cancer, so it's seen in 5 to 10%. So the KRAS is really huge, almost close to 50%. The BRAF is seen in 5 to 10%, and in the same way, you cannot treat this mutation with the cetuximab and the panitumumab. They're not going to be effective. Again, the reason why is because BRAF is downstream of the epidermal growth factor receptor, but you can treat it with a BRAF inhibitor, and it's called the vemurafenib. Vemurafenib. That's so dumb.
That's cool.
I'm so dumb. Okay. Okay, so cool. Okay, so here it is right here. So the KRAS will act on the BRAF, and the BRAF will actually lead to proliferation. But if this guy becomes autonomous, so if you get a mutation in this guy, then this guy can, he can do his own thing, and he can just be, autonomous from the rest of these guys. So using this panitumumab and cetuximab it's of no use. Cool.
So why don't you use that from the start?
Because you don't know what is mutated. Remember, the mutation could be in the KRAS, or it can be in the BRAF, or it can be because you have too much of the receptors. So like, if you have colorectal cancer, right? You've had a series of mutations that led to that colorectal cancer. Now, if two people have colorectal cancer, now it's not the same types of mutations that produce that colorectal cancer. So you see what I'm saying?
Yeah.
So you have to know like what is actually like mutated.
Oh, so that BRAF inhibitor isn't full.
It's only when BRAF
It's only when
autonomous
BRAF
on itself.
is mutated. So like these are just possibilities of mutations that can occur that can lead to colorectal cancer. And depending on where the mutation is, then you treat it at that level of mutation. Does that make sense?
Yeah, I think so.
A little bit?
Can I see the picture again?
Yeah, sure. Let me just remove all this. Alright, so you can have like a patient that comes in, you have like one patient comes in and they have a mutation associated with this receptor.
Yeah.
When there's a mutation associated with this receptor, it just means that this receptor is being overly produced. Alright? Or you can have a patient that comes in and they have a mutation in the KRAS. Or you can have another patient that comes in and they have a mutation in the BRAF. So these three patients have mutations at different points, but at the end of the day, they're all leading to proliferation. So the thing is that you want to target, depending on the mutation the person has, you want to target that mutation specifically. That's what I'm saying?
Yeah. So if there's a KRAS mutation, what do you use?
Yeah,
Okay, if there's a KRAS mutation, so I didn't see anything on the slides.
Yeah, I don't
That's
remember
why I didn't.
going
That's
over
why I
it.
didn't put anything. I didn't see anything on the slides. I actually Googled it before I was coming here, but I ran out of time.
That's real.
So, okay.
So you said the BRAF mutation is seen in about 10% of.
Yes, 5 to 10%. 5 to 10%. Okay, so I'm almost done. I just got one last thing to talk about. Okay, so I think this will actually help to tie in with the why the title of the lecture is what it is.
The last one.
Okay, so let me see. What's the title again? Okay, so precision and personalized medicine. Okay, so I guess the idea that he was trying to establish is that because of the fact that cancer tumor is caused by a series of mutations, when you go about treating someone that has cancer, you want to get an idea of the kinds of mutations they have so that you can give them a personalized, precise treatment. Does that make sense?
Yeah.
So that's how it's
Because
tying in with.
the cancer itself is individualized, so you want to make individualized treatment.
So, but overall, I still think it's not a good.
We barely talked about it, so I get you.
Okay, so the idea is when someone has cancer. Okay, so the old way of how they did things was that when someone has cancer, you just figure out, okay, they have breast cancer, they have colon cancer. You give them a generic drug, all right? And then you say, okay, this person is a male, he's 30 years old. This is the amount of drug we give to 30-year-old patients. So you give them a generic drug for the cancer, and then you give them a certain amount based on, what has already been established, right? But the idea is that being general or doing this is not going to work for everyone, right? So like the cancer that is occurring in one person, so the colon cancer that we see in two people, they are not, it's not all coming from the same mutations, right? So if we treated everyone generically, right, it turns out that only some people will benefit. So a certain segment of this population will benefit maximally. So the people that actually fit, it's kind of like, you're watching TV and then you see all the people that are lean and they're fit and everyone looks good and everything like that. But then like when you step outside, you got people overweight. So I mean, there's some people that, I mean, like they look like that. They look like what we see on TV, but it's not everyone, right? So I mean, it will work for some people, but then it's not going to work for everyone, right? So so this is essentially the old way, right? The doctors they get the biopsy of the tissue. Oh, so they see, this person has breast cancer. They have prostate cancer. I can see it because I'm looking at it under the microscope. I see that it's breast cancer, prostate cancer. So now I give them this drug X, and I give them this amount of drug X because they're this age, they're this size, whatever. Use the formula. But the idea is that when you look at the population of the people, all right, there is only a certain group of the population that the drug, number one, is going to be beneficial to, and it won't be toxic to them. All right. Then there are others that the drug will not be toxic to them, but they're not going to benefit. Then there are those that the drug will the drug is toxic to them, but they will benefit. And there are those that the drug will be toxic to them, and then they won't benefit. So they lose. Completely lose. Shut down. So okay, so then now it's important now that we start to use this new way. So we can get a sample of the patient's blood. We can extract the genomic and proteomic information, and based off of this information, then we can tailor the type of medicine we give them, and then the type of dosage that we administer to the person. So maybe based on the proteins, now we kind of have an idea of how much of this enzyme, the enzyme that degrades the drug, how much we should give, something like that, Does that kind of make sense? So this is the new way. That's the old way, and this is essentially what the this is it for the lecture.
I have a question.
Yeah.
It's not directly related to this lecture, but could you quickly go over microarray? Like what exactly goes on there? And what it
No one knows.
right. Because no one's been able to I mean I'm not
Yeah.
probably didn't go over, but if you know.
Oh, for like
So say you have like a million wells or something.
Right.
And within those wells you can test, so say you have I don't know, so you have like this string of DNA that has like all these different genes. So you can put like a piece into each
A piece of the different genes.
yeah, so like
Okay.
different gene coding for different proteins. So
Okay.
say you have like 100 different proteins and you want to see if these certain proteins are expressed under certain conditions. So you'll put the 100 DNA pieces encoding for the protein into your 100 wells or whatever. And then you could subject that microarray to like varying temperatures or like any other conditions that you want. And then from
to see if
there,
it's still expressed.
yeah, from there, that's where you get the different colors because you can see the expression levels under whatever conditions you subject them to within that microarray. Or you can do like, I don't know, like you can do microarray with like different bacteria and different like media. You can do it with anything. So it's basically just a way to test conditions for a lot of different like genes or proteins or whatever.
And stages of development.
That too, yeah.
Perfect.
You got empty PhDs?
You're an empty PhD?
Okay, so maybe just ask you guys one or two questions, and then we can call it a day.
Oh, I have one question.
Yeah, go ahead.
So we talked about how I guess the cascade is like all the way down, and with the drug vemurafenib, right? And it's more downstream than most of the other drugs in a sense. So technically, couldn't they just have given someone the vemurafenib, and then that could have pretty much helped them because it's like stopping that, I guess, that cascade.
I see what you're saying. That's actually a good point. However, I would also add that remember that tyrosine kinase is not just acting only on the RAS because there were other adjunct proteins that were getting phosphorylated that were leading to. Let me show you. So here you go. See how complicated this is. See that, right?
Yeah,
So
okay.
the fact that it's going through this whole, let me see if I can get rid of this. The fact that it's going through the RAS, GDP, GTP, that's just one thing. It can go through this pathway as well. So it would still have an effect, right? But it doesn't mean that you're not still gonna get, like I guess a tumor cancer formation and stuff. All right, just a couple questions and then we'll call it a day.
Yeah, it was supposed to rain today. It did rain today.
Yeah, there was supposed to be a flash flood warning from 4 to midnight. Okay.
Alright, so you guys can speak to the person next to you.
So that was the first one. This is that was the KRAS.
That's the main retinoic acid.
That's right. That's right. That's very good.
Folic acid. Or no, FAD and NAD.
Yeah.
But the percentage was.
I looked at it. I didn't get actual.
Okay,
I'll look at it. I'll tell you today. I'll tell you. Oh yeah. I'll tell you. That's right. I'll take you. I'm not gonna remember this. Shout out.
Am I putting this right? Yeah.
Snap.
know.
He speaks medical medicine language.
Oh dang. Okay.
I don't know why, but DC only rains at night. I've never experienced rain in the middle of the day. Oh, yeah, that one.
Maple syrup.
Can I put that?
Oh, that's
Yeah, two-
like every time I wake up, it's just wet. I'm like, oh, it's outside, it's wet. I guess it rains.
Is another wrench chain?
All right. So another question for you guys.
Another question.
What are these acting as?
Inhibitors. Competitive
Competitive inhibitors in what sense?
They protect
Antibody binds to the receptor and blocks it.
Okay. Okay. Monoclonal antibodies.
Monoclonal.
Okay. Monoclonal
Say that
antibodies.
again?
The monoclonal antibodies.
No, what does that have to do with the Centennial?
What does that?
There. So
No, I just sneezed. I sneezed and then it was like at the same time. That's how I sneezed.
so this is a protein, right?
And then it came
So
out.
your antibody binds to the protein. Right, so it's specific to a certain protein. So these drugs here, they're functioning as an antibody.
Was that the last question?
Yeah. We'll call it a day. Actually, there's one last thing I want to point out to you guys. Just one last thing. So your epidermal growth factor receptor is made of proteins. So your cetuximab and panitumumab, they're monoclonal antibodies that will bind to this receptor and then block the binding of the epidermal growth factor.
So they both bind at the same site? Is that why they say that it's competitive?
It would bind to the site where the epidermal growth factor. Exactly. That's right. One last thing that I'll point out to you guys before we head out. Where did it go? Okay, so when you're looking at colorectal cancer, so the first mutation that usually occurs is your APC gene. All right. Now, APC is a tumor suppressor. Remember that was with your E-cadherin and your beta-catenin, right, which was holding the cells together. So now there is a condition, a genetic condition where this gene here is mutated. So it's a genetic condition. This is your familial adenomatous polyposis. I believe it should be autosomal dominant.
Yes.
It's dominant?
I think so.
Yeah, I think it's autosomal dominant.
It's on the slides.
Oh.
All right. So
Check what it is.
if you have this inherited condition, that means that you're gonna have polyps in your colon. So you're already gonna have this first mutation present. But generally, when you have colorectal cancer, the first thing that does get mutated is the APC gene. Right? But you can have an inherited condition where the APC gene from the get-go is mutated, which is your FAP, your familial adenomatous polyposis.
Oh, yeah. I do remember that. I just remember FAP.
And you guys remember that the polyps are benign. So like your adenoma, the polyps are benign. They're precancerous, but they're not the.
It can mutate to be cancerous.
Exactly. More mutations you need more mutations before